Okay, so the paper that we're doing today is thalamic activation of the visual cortex at the single synapse level by Chen et al. It is it's a very recent paper. So just a little bit of a background for just to get us oriented here. So the input from the retina goes through the optic nerve, and its first location that it goes to is the lateral geniculate nucleus of the thalamus. Cells in the lateral geniculate nucleus of the thalamus then project to the primary visual cortex. This paper is going to be talking about the primary visual cortex of the mouse, specifically layer 4 cells in the primary visual cortex of the mouse, and how their receptive fields are built out of inputs from the lateral geniculate nucleus. So if we look over here, you guys can see my mouse?
Yes, we can.
Great, thank you. So if we look over here, there's kind of a layered structure in the lateral geniculate nucleus that gets different eyes, different from the right and left. We don't care about that right now. There are the axons from those lateral geniculate nucleus cells are going to come up and innervate the layer 4, mostly the layer 4 of the primary visual cortex. And there is a very old model that this paper just essentially nails. The receptive field properties of these lateral geniculate neurons are what they call center surround. So there's each cell in the lateral geniculate nucleus is going to be looking at a particular location in visual space. It's going to, if it's an quote unquote on-centered cell, then it's going to activate when a bright thing is flashed in the center of its receptive field, and it's going to be inhibited if that white is flashed the area immediately surrounding that center. So that's an on-centered cell. It has an on-center and an off-surround. There's other types of cells that are exactly opposite of that. They're called off-centered cells. If a light is flashed on the surrounding area, then it activates, but it's flashed in the center, it gets suppressed. So those cells are projecting to the to layer 4 neurons, and those layer 4 neurons usually have a receptive field that is looking for edges at a particular orientation. And Hubel and Wiesel in this 1962 paper discovered this, and found out that that these visual cortex neurons in the cat, in this case, were selective to orient a particular orientation, and they came up with a model that said, hey, we know that the lateral geniculate nucleus has these cells that look for little dots, essentially. Maybe the cells in layer 4 or the visual cortex that look for lines or edges are really just collecting synaptic inputs from a whole bunch of LGN cells that are in a line. And that was that they proposed in this 1962 paper. That you will see is 100% correct. And whereas there's been many papers through the years that have bolstered that idea, this one actually does the best so far in just nailing it. Okay, so let's go to figure 1. Everybody still on with me?
Yep.
Great. Okay, so so the first thing they did is they used a calcium indicator, Cal 520, to label all of the neurons in layer 4 of this mouse, so that they could look at their orientation selectivity. So here is the mouse. It's looking. It's there's a two-photon microscope that's looking at layer 4, deep into its visual cortex. It's being shown grading patterns. Those grading patterns are going in different directions. And so if you look at C here, you have a grading going to the left, and then you have a grading going to the upper left, and then you have a grading going to the top. And this is showing for each of these individual cells what the responses are. And so we can see that cell 1 here likes things that are edges that are going to the bottom right.
Can just
Yes,
to ask a quick question. So these are drifting gradients then?
These are drifting gradient gradings. They don't make a big difference between orientation sensitivity and direction selectivity in this paper. So in fact, they don't really talk about direction selectivity per se. They just talk about orientation selectivity. So so they're using stimuli. It's a good question. They're using stimuli. And by the way, please, if I'm saying something wrong, please stop me. My reading of the paper is that they are ignoring the type of temporal aspects and really trying to look at just orientation selectivity. But they're using grading, drifting grading, so they're showing plots that clearly show direction selectivity as well. It's okay, so they did some calcium imaging. They did some whole cell recordings of the exact same cells. Not a big deal. Really, what they did this so that they could find cells that were highly orientation selective, and so that they could take one cell and electroporate it with a calcium indicator. Okay, and so in this case, they were using JG-Camp 7 something, JG-Camp 7B. Okay, so this whole neuron now is this single whole neuron is going to be expressing this very sensitive calcium indicator. And the reason why they did that is because they wanted to look not only at the cellular response, but look at all of the dendritic spines. And I'll show you that in a second. But the other thing that they did is they monitored this particular cell over a full month to make sure that its orientation selectivity was constant over that time, and it looks to be constant over that time. So, like I said, they put in this calcium, this genetically encoded calcium indicator, GCamp, so that they could look at individual spines. So now they're zooming in on one of these, this particular cell, this particular layer 4 cell that we already know the direction tuning of the whole cell. And now they're looking at these individual spines. Each one of them is supposedly going to show a calcium response that is mirroring the presynaptic cells firing. Now we'll see at the end that that's not actually true for all of these, but the initial assumption was that it would be true. Okay, so if we look at, let's say, this spine number 1 here, that spine is showing a response if the gratings are moving to the upper left, and so that means that whatever neuron is giving rise to that synapse has a receptive field that is orientation selective and direction selective as well in this sense. Case. If we look at synapse number 2 here, we see that it is also responding, but is responding differently, right? Because it's coming from a different cell. We don't know what cell, where that cell is right now, but it's coming from a different cell, and it's responding to gratings that are moving either to the left or to the right. And so this would be a non-direction selective, but an orientation selective cell. And so they can look at that for all of these synapses on this neuron. And one of the key things that they did in this paper is they really did long multi-day recording sessions where they could look at essentially all of the spine, so they could get really good statistics about what's going on. Okay, so they could look at all these spines, and what they found, which was very bizarre, is they found that there was no evidence of calcium transients that were expected to arise from the activity of these canonical lateral geniculate neurons, which are supposed to be active at every orientation of the visual stimuli. So let me just go back here. What they should have found is they should have found the lateral geniculate neurons that are known from other studies to be making synapses on these layer 4 cells that are orientation selective. Those are supposed to be giving, they are supposed to be looking for dots. And a dot is going to be not caring which direction these gratings are going. They should pulse every single time that the grating goes past. And what they found is that none of these spines that they were looking at with this calcium indicator was showing that, which is astonishing. Okay, and so it probably makes sense for me to just say what happened. It turns out that there's that the spines, I'm going to show this later, but I don't want to confuse people. The subset of spines that are getting their inputs from the lateral geniculate nucleus look like they do not have calcium responses. They are fine synapses, but they don't actually have calcium responses, which implies that they don't have NMDA receptors on them. They just have AMPA receptors. NMDA receptors are the ones that actually open up and allow calcium to come in every time it's a presynaptic glutamate that is put on the spine.
Can I ask a quick question?
Please.
So okay, so the spines that we're able to see with calcium, because I know where the rest of the paper goes, but I don't want to give it away.
Yeah. You can give it away. I mean,
it's okay. It's okay. We're
not trying to like confuse people.
Yeah. So these are not showing any untuned, so not orientation selective responses. It's pretty much all orientation selective, and this means that whatever may be providing what Hubel and Wiesel actually required for their model, if that were true, that would be coming through something that doesn't have the CA2+ response. And does this also mean, because this is the NMDA receptors, that the connections that would be providing this non-tuned input, that they would not experience, say, LTP, for example?
I believe that is the case. So they in their discussion section, they reference a paper that talks about non-typical learning in these synapses, and I did not look up. It was on my to-do list, but I didn't get around to it.
Yeah, but that was the takeaway that I was kind of going towards. I was thinking, does that mean these are fixed in a sense? Yeah.
And it might be a developmental thing. Maybe they did. This is this was an adult mouse.
Yep.
So so at this point in the paper, though, they don't know what's going on. They're like, wow, there's supposed to be these inputs. We don't see those inputs. Maybe. I doubt that they were thinking this way, but you know, they in the paper they're definitely thinking this way. They say, well, maybe these are coming from the subset of actually direction selective cells in the lateral geniculate nucleus. So an extra complication, there are some cells in the lateral, in a particular region of the lateral geniculate nucleus that actually do have direction selectivity. So, so what they did next. So let me just read this. So the paucity of canonical inputs could be the consequence of a preponderance of thalamic orientation-tuned inputs. So maybe there are orientation to maybe Hubel and Wiesel are wrong. Basically, is what they're saying, right? We therefore examine the presynaptic connectivity of the orientation-tuned L4 cells morphologically to assess the proportion of inputs arising from the LGN shell region. They used a transsynaptic rabies virus, and let me show you that. So we're moving to figure 2. So they took one of these cells in layer 4 of visual cortex, and that they knew was orientation selective, strongly orientation selective, and they injected it with a rabies virus. The rabies virus is going to label all of the presynaptic cells to that single cell, which is really cool. I mean, this is it's like this is a single layer 4 cell, and it's showing all of the cells that are connected to it. So these are the 2 critical slices here. All the cells that are lighting up are the ones that were presynaptic to this layer 4 cell. Most of them are in V1, right around the cell. There's a few in a higher order visual area called LM, and here is the ones coming from the dorsal lateral geniculate nucleus. Okay, and this is zooming in on each of those. This is a layer 4 cell that is being targeted. So you can see that there's a whole bunch of cells within layer 4 and layer 2, 3, and even layer 5 that are presynaptic onto our little target cell. And in lateral geniculate nucleus, there's a whole bunch of cells. They go and they make a 3-dimensional map of the cells in the lateral geniculate nucleus. And they do this in order to determine if those cells might be in the location that they know is containing the direction selective cells in the lateral geniculate nucleus. Turns out it's not. So that was their way of saying, nope, it ain't that. It must be something else. This is just a figure that is showing the percentages of cells that are presynaptic to this target cell. Most of the cells are within V1 itself. Only about what did they say? 9. I don't want to say that. There's only about 10% that are actually coming from the lateral geniculate nucleus.
I think it was 73% in V1.
Exactly. I find this I already knew this. I knew this. It says this in all the textbooks, but you know, it is always surprising to learn that the actual input as opposed to recurrent connectivity is only about 10%. So the vast majority of stuff that's happening in the cortex, even at this layer 4, is recurrent connectivity. And it's really the only connection to the eyeball is through this LGN 10%. Okay, so they say the vast majority of the thalamic cells were in the ventromedial region, which is in the LGN's core, a region harboring non-tuned canonical LGN neurons. Thus, the experiment ruled out the possibility that the highly orientation-tuned L4 neurons are predominantly connected to orientation-tuned LGN neurons. Instead, the experiment raised the possibility that most of the thalamocortical inputs do not produce a postsynaptic calcium accumulation. Okay, so this is they're now saying, okay, this is probably that they just don't show up in a calcium indicator. So we're moving on to figure 3. So what do you do if you can't record calcium responses in your dendritic spines? Well, thank God, a few years ago, people invented something called glue sniffer, which is a glutamate. It's a fluorescent indicator of glutamate itself, right? So instead of looking for calcium transients, they can look for actual glutamate being squirted onto these synapses. Okay, so so in this experiment, they do a couple of things. One is that they take a single layer 4 pyramidal neuron that is orientation selective, and they inject, let's just say inject. They inject so that it expresses this glutamate sensor. Okay, so that should, unlike the calcium indicator, that should give a response to any excitatory input, even if it doesn't have NMDA receptors. Okay, and so this is in B here. This is the cell that they transfected, and in C, this is looking at one of the basal dendrites, highlighting particular synapses that they are doing glutamate sensing on. And they found, and the other thing they did, which I'll ignore I'm going to look at column D here for a second. So they found that this comes in 2 categories. Okay, there's 2 different types of synapses. There's orientation selective synapses, like that we had already seen before. So if you look at, let's say, synapse 3 here, it is liking gratings going to the right. And then there are the non-selective ones that I talked about that will fire no matter what direction the grading is going, because these are the on-center or off-center ones that are supposedly coming from the LGN. So we finally are seeing the types of inputs that we expect to be there by using this glutamate sensor. But they wanted to make sure, okay, are these synapses that are now showing this, are they actually coming from the LGN, or maybe they're coming from somewhere else? So what they did is they used a couple of different techniques to silence the cortex. Okay, the one that they're showing here is an optogenetic technique. I'm not going to go to it, but when they silence the cortex, silence all of the cells that would be generating excitatory input from the cortex onto this cell, these responses go dead. But these non-selective responses are unaffected, and it comes back just fine after you unsilence the cortex. So right there, they've proven that that the synaptic inputs from the cortex are the ones that are orientation selective, and the ones that are coming from the lateral geniculate nucleus are non-orientation selective. Any questions? That's kind of an important thing to understand.
No, I think we're good. I just thought this was crazy when I got here. I was also surprised by how impressive it was.
This is why I picked this paper. It's like, so it's such a strong result. Oh, the other thing, this is like the level of the result here. It's like silencing versus no silencing on the orientation selective versus on the non-selective. It's like it's rare you see like a neuroscience paper that is so absolutely clear in its results. But so what's important about this? One of the things that's important about this is that now they have a way to distinguish what's from the thalamus, what's from the lateral geniculate nucleus, and what's from the cortex, which is going to come in useful later on in the paper. The other thing, this is a supplementary figure 5. They could also take a look at these at individual synapses and do white noise stimuli so that they could actually plot the receptive field properties of these cells. So for instance, synapse 1 here, these are the responses to kind of flashing black and white dots. And if you use those to plot the on portions of the receptive field and the off portions of the receptive field, each of these green squares is the visual field. This is a 10-degree visual field scale bar, and so you can see that these have the classic on center. Orange is saying it's active there, and a blue surround and an inhibitory surround. And then there's other ones like this other synapse here that's an off center. It has a blue center with an orange surround. So these are behaving just like what we would expect classically the lateral geniculate nucleus cells to behave. And one of the things that they did here is they in a different experiment is they find in the same experiment in the same cell the glutamate sensor iGlu sniffer 3 with the calcium indicator RGECO1A and so they could look at these purported corticocortical synapses that were orientation selective, et cetera, and they could see that every time that they saw glutamate presynaptic activity, they would also see a calcium transient. So those are tracking, but with the thalamocortical synapses that don't have an orientation selectivity, they wipe out. There is no calcium response that is seen. So it's a very clear indication now that these thalamocortical synapses simply don't have a calcium response, supposedly because they don't have NMDA receptors. Okay, moving on to figure 4. How are we doing on? Okay, so the successful identification of thalamocortical and corticocortical excitatory synapses allowed us to build up a functional map of nearly all layer 4 dendritic inputs to a single orientation tuned neuron. And in order to do that, they needed an experiment that lasted several days. So they talk about figure 6 here. Forget about B, but in order to do these experiments, to map essentially every synapse, they had to take multiple field of view so that they could zoom in on little pieces of the dendrite and go through the visual stimuli and record those then move to another visual area, and another part of the dendrite, another part of the dendrite, another part of the dendrite. And they did that over multiple days. This is showing day 1, day 2, day 3. So a quite impressive experiment. But what they got out of is they got a really comprehensive map of essentially all the synaptic inputs, maybe like 90% of the synaptic inputs. And they could classify them into these two types. So this is from, again, from a glutamate experiment. Here is this single cell. It's direction selected. So if you look over here, it says the axon. That's the cell's own direction selectivity. Okay, its own orientation selectivity, and it likes gratings that are going to the right. That's how to interpret this. Likes gratings that are going to the right. The thalamocortical synapses are in pink here. You can see they're all over the place. They're not clustered or anything. They're just wherever the axon from the LGN passed the dendrite. And all of them have responses that are not orientation selective. So it doesn't matter which direction the grading is going, it responds fine. But if you look at the corticocortical synapses, which are shown in C here and blown up in D, those have a preferred orientation. And so these are some of the corticocortical synapses that are shown there, their responses. And they've labeled it in such a way that if they like gratings going to 0-degree direction, then it's orange. They like it in the 90 directions, it's blue. So that's what the rainbow means. And the size is the overall direction selectivity. I'd have to look that up. I think it's not the size of the synapse, it's the size of the response that is being seen. Couple things that I want you to get out of this. If we look at at all of the histogram of the corticocortical synapses, all of these that are, and look at their individual orientation selectivities, they show that the majority, the mean of those is in the 0-degree direction, which is exactly what the cell is also showing. So we've seen that in several other papers, but you can predict the response of this cell based upon which corticocortical synapses, which neurons it's connected to. It's kind of the average of all the of their orientations selections. The other thing to note is that there is a clustering trend. So if we look at the corticocortical synapses and we plot the correlation of those tunings, okay? So if I take any two synapses, like one is going to that are corticocortical, they're going to be orientation selective. I can compare angle, and that gives me a correlation. So if they were right at the same angle, then this would be a high correlation. If they're 90 degrees off, then it would be a low correlation. Okay, and so this is plotting the correlation between pairs of synapses as a function of the distance between those synapses along the dendrite. And so this is showing that there's a peak over here that's if two synapses are within 5 microns of each other, then they're more likely than chance to have the same or have similar orientation tunings. Again, we've seen this. It probably says something about how learning occurs. There's some kind of local learning that is enhanced that makes one synapse enhance others that are firing at the same time.
Yeah, we've seen this in previous papers, right? That we presented here.
We have
Yeah.
definitely seen this. I think one of the papers that that you gave, Randall, in Hippocampus was showing clustering as well.
Yeah, again
Right.
with that same pattern. Yeah, but I'm actually super impressed with the very even distribution of the TC synapses. That's almost surprising.
It is, and they didn't say that there was any clustering of those, which I mean, there's no orientation selectivity. I guess they could have said maybe on center versus off center could be clustered. And in the figure 5, at least I don't see any type of clustering in this. So this is the final figure of the paper. So what they did here is they used gratings, okay, and so they could definitely look at orientation selectivity and everything, but they couldn't look at the position of the thalamocortical input. They couldn't look and say this thalamocortical input from LGN is looking at this point in space, right? And so what they did in figure 5 is they did the same type of comprehensive experiment, but they're only doing this now on the thalamocortical synapses, and they're using stimuli that are a single bar moving across the screen. It can either be a white bar moving across the screen in some direction, or a black bar that's moving across the screen in some direction. And by combining the responses of, let's say, this particular spine that's getting an input from the LGN, they can notice that the receptive field center of that LGN cell that's presynaptic here is right there in visual space. And this the off surround is around that. And so they can essentially, by using these stimuli, say exactly where the center of the receptive field is for each LGN input, and they can say whether it's an on center or an off center. They do that for all of these approximately 500 synapses onto this target cell, and then they plot all the on centers and off centers in visual space. So here's our cell. This is like the beautiful figure of the paper here, right? So here's a single cell. We're looking at only the synapses that are coming from the LGN. We're labeling, we're coloring the on center ones red, the off center ones blue. I don't see any clustering. Maybe there is, but I don't see it. And then we're plotting in visual space each of those synapses where its center of its receptive field is. And what you get is there's a whole bunch of these on center ones in a line, and it's flanked by off center ones also in the same line, which is exactly, if you recall, what Hubel and Wiesel model was proposing. Does that make sense? This is kind of the key figure here.
Yeah, it's a great result, great validation. Okay,
and just as like a real takeaway of this, this is making it really clear how function gets built up from structure. Yeah, this is like predicting a ton of stuff about the receptive field property of this particular neuron, based upon the receptive fields of all the inputs going into it. And did this for 6 total neurons. Each of these is like a crazy long experiment, multiple days. So they did this for 6 neurons in 6 different mice. And each time they looked at the measured layer 4 neurons orientation selectivity versus the orientation selectivity that could be gathered based upon plotting this of all the inputs, and it's dead correlation, which totally makes sense. Okay, so a couple of the main results of this paper. I guess the astonishing part of the paper, the unexpected part, is that overall our results are consistent with a reduced or absent expression of NMDA receptors at thalamocortical input receiving spines. And this is where it says, additionally, the absence of postsynaptic calcium signaling at TC inputs provides an explanation for the observed unconventional plasticity rules at TC synapses in V1. So like I said, I haven't looked up this reference 47. So that was surprising. And I feel like, how can we be in 2026 and not know that the basic synapse complement of receptors on one of the most well-studied synapses in all of neuroscience? It's just kind of amazing. Anyway, the other thing is our approach enabled the identification and characterization of nearly the complete set of active LGN synaptic inputs to a single orientation selective L4 neuron. The spatial aligned receptive field centers from LGN inputs matched the preferred orientation of the corresponding neuron. Thus, our findings directly validate 4 predictions of the connectivity proposed by the Hubel and Wiesel model. And that's all I got. So I'll open it up to questions.
That was a fantastic paper, by the way. I loved it every bit of the way. The only thing that I now wish,
nominated for an award.
Yeah. But still, there's one thing that I would really like, because you remember the award is actually meant for static data. So I would really love it if the Allen Institute's upcoming whole mouse brain results make it possible to identify the receptive fields in DLGN and trace connectivity to L4 and give the same result purely based on static data, even though that still wouldn't be able to pick up on the non-NMDA nature of those DLGN connections.
Let me double check that I understand. So when you're saying static, I mean they have calcium imaging of individual cells.
Right, but they're still doing, I mean, I guess, okay, we can quibble. I would love it if it was just gotten straight out of the structure directly and not having to observe the behavior directly. Of course, you want that as well. You want this part of the study too, because you always want the, let's say, the structural microscopy, perhaps EM, and you also want the calcium imaging, or in this case, glutamate imaging, and you want some kind of other behavioral, say, result or model or something like that. Be nice to have all of them.
Yeah, I mean, I think this talks to the idea that if you only had structure, that you could figure out a lot from only having structure.
Yeah, you could probably figure out how it works, except you still wouldn't pick up on that NMDA, the missing NMDA receptors, I think.
Yeah. Ariel?
Yeah, perhaps the computational modeling minded people can give a clear answer to this. But I'm just thinking, if these cells are just inheriting their orientation selectivity from their inputs, and that's like 75% or something of their inputs or so, like, where do they get their, like, where does the original orientation selection information come in from? Like, if they're...
But what this is saying is that if you silence the cortex, you will still have orientation selectivity based upon the fact that these thalamocortical inputs are in a line. So it really is that, you know, it's this 10% of LGN inputs that are spatially laid out in a line that is the basis of orientation selectivity.
Yeah. So
And
even
then
if
that
even if the
enhance-
individual
I
LGN cells
misunderstood figure
themselves are not
5.
orientation selective. Yeah. Yeah.
Sorry, no, that makes perfect sense. I just misunderstood figure 5 when it was talking
Well-
about specifically thalamocortical inputs.
And it has been a back and forth in the field, at least a few papers that have argued that that it might be coming from these recurrent connections. So this puts that theory to rest, I think, if any neuroscience theory ever gets put to rest.
Well, the recurrent connections still amplify the effect, right?
Well, let's see. So the one we did 2 weeks ago was arguing that those were doing more than just amplifying, right? That they were trying to predict into the future. That included a whole bunch of other stuff. But I think at that point we have to first of all, these are layer 4. These are layer 4. They're simple cells. So whereas they might be orientation selective and they might be direction selective, they're not going to be how do you say it? The receptive field is not going to be agnostic to where the edge is.
Right.
There's some more complicated stuff that's happening, and you saw there's feedback from higher visual area onto the layer 4 cell. This is the other thing that's really cool about these papers. It's like, just nature is wiring stuff up with a casual disregard of the blueprint. It's like, oh yeah, we should mostly have feedback going to layer 2, 3. But if you run into a layer 4 cell, go ahead and make some synapses. I'm not going to care. Whatever learning algorithm there is, I'm going to roll with it.
The other question I have, which is, do we have any idea why these thalamocortical synapses don't have NMDA receptors? Like how actually that works? Like I'm thinking about it from the V1 neurons perspective. So like it doesn't know where these synapses are coming from, whether they're from another fellow V1 or from LGN or from wherever. Like how's it doing that and selecting?
Yeah.
Nicholas, if you know the answer, then speak up. Otherwise,
I would assume that it's that there are signaling proteins on the thalamocortical neurons that say drop your NMDA receptors, but I have no idea. Nicholas?
I remember, I kind of have a fuzzy memory from an undergraduate neuroscience class I took, and we spent a little time on the connection between retinal waves and how that led to the edge, the structure which informs function of the thalamus, in with the retina. And I remember being told, that there is NDMA expression early in the thalamus, and that helps to cause the weights, and then it stops having NMDA expression and just has AMPA expression. And there's a specific name for that, for a cell that has. Am I getting fact checked? Oh no. Okay. There's a
critical
name
period
for a cell that has non NMDA expression, but I forget what the name is. And I also remember being told that it's known that the feed backward, the visual cortex to thalamus, or was it? I think it was actually about the auditory system, and it was that you have increased sensitivity at lower levels that is caused by higher level feed backward, and that helps but that's probably already known to people here, and
Yeah, I wish I knew more about how this fits into the overall learning literature, because there's been a ton of experiments. And I didn't even look. I assume these are adult mice. I'm sorry, I didn't even look.
I'm just thinking again from the context of a memory experiment or a structure to function experiment. So yeah, I guess we start with the thalamus. We say like here's where the receptive fields in the thalamus come from, let's say, from the DLGN. Maybe we allow that as a given, like they've mapped that out with calcium or other sort of experiments. And then yeah, the idea is if they repeated this sort of figure 5, they built up figure E by tracing those connections somehow. And instead of doing it via these sort of glue sniffer calcium experiments, they did it just via structure. Do we think? I mean, yeah, I think that would win a memory decoding prize.
So, yeah, I think so. One of the things that I would love to see, this is somewhat an aside, but this paper gives as much statistics as we need to know about these thalamocortical inputs. If we were to model, actually Anton Arkhipov models these things, right? He models the statistics. Like imagine that we have a Python program that will generate L4 cells, and they will build up their receptive fields, just like is shown here, with on-center and off-center, with these types of random offsets based upon thalamocortical inputs from LGN. Now, let's say we had 1,000 of those, and we knew what their orientations were, and we had 1,000 LGN cells that were feeding into those, but we didn't know what their receptive field properties were. It's not clear to me that we wouldn't already have enough information to back calculate what those LGN receptive fields are, even though there is no way to calcium indicate them. And this is a fully computational experiment to say, can we use calcium indicators over here and knowing the connectivity to predict stuff that we don't know upstream in this? I think it would be a wonderful kind of computational experiment to do. Something a carbon copy should.
Yeah, I was already thinking about this, because you remember how I said we should make on our platform we should make examples for each one of the studies that we think are a good example for the prize. So this came to mind in essence. Yeah.
So
just a note.
You're noting the same thing here.
Yeah, because I messed up somehow the meeting link. We're in the wrong calendar event, which just means this one will end in 3 minutes. So if anyone has anything they really want to say, we should get it out now. Or not.
We have a paper for next time.
So I'm thinking of presenting next time. I haven't picked the specific paper yet, but I'm thinking of either doing one on grid cells or doing one on the mushroom body of the for fruit fly of Drosophila. One of the two.
Awesome. I love both of those.
Yeah. Applause to the authors of the study and to Ken for presenting it so well.
Thank you for the great presentation. And indeed, this is not just interesting as a paper, but this is actually something we can build a model around to demonstrate. Yeah. It's really cool.
Okay. Thanks everybody.
Thanks for sharing it, Ken. Thanks everyone for coming. See you guys next time. Bye.
Thanks, guys.